System divided by 2. System divided by 2. Hey, it's like that equals.
It's getting grave.
And then I add them together.
Questions? Come on, fire up.
Fire up.
No, you have to ask a question.
I was
Fire
going
up.
to say
Okay, let's continue our discussion of virulence factors and specifically exotoxins. Several of the next examples that I'm going to give you of exotoxins fall into a group called AB toxins. And this particular slide right here illustrates what an AB toxin is and how it works. An AB toxin consists of two functional parts, the A part and the B part. Okay? B part attaches to a specific cell receptor. Remember that toxins are oftentimes tissue specific and are oftentimes named or categorized based upon the type of tissue that they affect. Neurotoxin, hemotoxin, etc. Once that B component attaches to a receptor on the membrane, it stimulates endocytosis. Remember what kind of endocytosis this is? Good. This is receptor-mediated endocytosis. Okay? So, the whole toxin is brought into the cell in a vacuole. And then once it's in the cell, the A portion is released from the B or binding portion. And then that A toxin is released into the cytoplasm where it has some effect. In this particular example, they said that this particular toxin inhibits protein synthesis. Okay? But it can have a multiple effects depending upon the type of toxin we're talking about. Okay? So, one of the AB toxins that we're going to talk about is the botulism toxin. What do you know about the botulism toxin? It is a neurotoxin, good. Anything else?
Comes from like bacteria that's in water.
Comes from bacteria. It comes from a species of bacteria called Clostridium botulinum.
It used to look good.
Pardon me?
It used for Botox.
Yes, actually, that particular toxin used in tiny amounts can be used to treat wrinkles. Does anybody know how it treats wrinkles? It paralyzes the facial muscles, particularly in the forehead, that are responsible for wrinkles. So when you relax those facial muscles, the wrinkles temporarily disappear.
Since it works on nerves and muscles, does it inhibit calcium ion channels?
See in just a second. As it turns out, it's not calcium ion channels, but we'll see. The botulism toxin causes what we call flaccid paralysis of muscles. Here's what's happening. Okay? Here is a motor neuron innervating a muscle. Normally what happens when that motor neuron is stimulated, it causes vesicles that contain acetylcholine, one of the neurotransmitters, to diffuse into the synapse. And then as the acetylcholine binds to the muscle cell, we get contraction. Okay? This particular botulism toxin blocks these vesicles containing acetylcholine from binding to the membrane and from being released into the synapse. So we don't get proper stimulation then of the muscle. Now, that's a good thing if we're just trying to paralyze facial muscles, but if we paralyze muscles of the diaphragm, for example, and rib cage muscles, what could be the result? Asphyxiation. You can't breathe, okay? Remember, those muscles have to contract to increase the volume of your thoracic cavity, which creates the negative pressure that causes air to enter your lungs. Where have you probably heard of botulism toxin in the news? It is a food contaminant, potentially. Someone said an organism that lives in water. Actually, it's more common to find Clostridium botulinum spores in the soil. Yep. They are anaerobes. They're strict anaerobes. Contaminated vegetables, for example, like green beans, could potentially be a source of botulism toxin if they are improperly canned. All familiar with the process of canning and what it entails? Basically, you take your vegetables and you put them in jars and seal the lids, put some liquid in there, and then put them in a pressure cooker. And cook them for prescribed amounts of time. What's our equivalent of a pressure cooker? Autoclave. Okay, so we're basically autoclaving those beans. Why are we doing that? Because we want to destroy any endospores that have remained, because little bits of soil, even if you wash the beans or the other vegetable, can still adhere to the outside and endospores can contaminate them. Okay? But if you pressure cook properly, then that will kill the cells if you reach that 121 degrees centigrade, 15 pounds per square inch for 15 minutes. Okay? One of the things that the process of autoclaving does, however, is it tends to force the gases out of the liquid in the jars that you're canning. And so that tends to become an anaerobic environment. So if you don't achieve the proper pressure and temperature, then the spores can survive canning, and then in that anaerobic environment they can germinate and produce the botulism toxin. If you ever see a can on the grocery store shelves, and it should never make it that far, but if you did, and that can was all distended and swollen, that would indicate that anaerobes had survived the canning process and were fermenting and producing gas. And if that anaerobes were Clostridium botulinum, then it could have botulism toxin. Go, good question.
Yeah. So is it the cans that you see on the shelf that are the cans that are being used to?
You can industrially cook things and pressure cook them in cans. Home canning usually is done in jars because the jars can be easily sealed and opened. Okay. So the canning is more of an industrial process. This is really a nasty toxin. Okay? Micrograms of this stuff can kill people. I don't know who did the calculation, but they said 30 grams of botulism toxin would be enough to kill every person in the United States. 30 grams. So one of the areas where you might have also heard about this is botulism toxin as a possible bioterror agent. If it could be introduced into a water supply, for example, it could be devastating. Actually, there are 3 categories of botulism. The one that we just talked about, foodborne botulism, improper canning. There is another type of botulism that's referred to as infant botulism. Infant botulism is oftentimes associated with kids under the age of 1 consuming unpasteurized honey. Honey has a lot of botulism spores in it. Okay. If you're under the age of 1, then you really haven't developed your normal flora completely, and therefore that antagonism that normally would suppress the germination of those spores isn't working properly. And so infants can develop botulism infections from eating honey. And so that's one of the reasons why they say don't give honey to a kid under the age of 1. You can also get botulism spores in wounds, kind of the same way you get Clostridium perfringens spores and gas gangrene, and that can also cause a wound infection, botulism. Another AB toxin that is also a neurotoxin is tetanus. If you were to ask the average Joe on the street what they associated tetanus with, what would probably be the association they would tell you?
Stepping on a nail.
Stepping on a rusty nail. Okay. And actually, there is some correctness in that perception, in that developing the disease of tetanus can really only happen if you have a puncture wound. Another species of Clostridium, nasty genus, Clostridium tetani, is an anaerobic endospore forming bacteria, just like its cousins. Okay? And if you puncture the skin and that thing that punctures the skin is contaminated with these spores, then they can kind of almost be injected into the lower tissues. The damage helps to kill the cells. So we create our little mini anaerobic pocket in those areas where the cells have been damaged and they're dead. And then those tetanus spores can begin to germinate, and the cell then begins to produce a toxin. It's a neurotoxin as well. The neurotoxin is called tetanospasmin. Now this is kind of an interesting neurotoxin, because it migrates down neurons and actually affects and inhibits a special type of neuron in the spinal cord. And the type of neuron that it specifically targets are inhibitory neurons. Remember those from physiology? Spinal inhibitory neurons. So if muscles are going to relax after they have been stimulated by a stimulatory neuron, an inhibitory neuron then has to basically send an inhibitory signal that causes relaxation of those previously contracted muscles. This tetanospasmin migrates up the nerves and to the spinal cord and specifically inhibits these inhibitory neurons. So we basically don't get the signal that says shut off contraction. And muscle contraction continues to be sustained and sustained and sustained. And it can be so strong and so sustained that actually can break bones. It can pull ligaments and tendons away from bone. That muscle just goes into complete, constant contraction. And it's not uncommon to see the arms arch up as a result of that, the back arch as a result of just these continued, sustained muscle contractions.
One question.
Yes?
What if you had like big biceps? If you use their flex like that?
Wouldn't you have big what?
Biceps?
I don't think you survived long enough to.
Because you relax.
Not a way I would recommend bulking up. Very high mortality rates, about 50% mortality rate. Mortality is usually due to respiratory failure again, sustained contraction of the diaphragm, the rib cage muscles, means you can't relax, contract, and get that positive negative pressure that you need to inhale and exhale. Now, if you can get somebody on a mechanical ventilator that can breathe for them, and then you can administer an antitoxin, then you've got a reasonably good chance of survival. This is one of those immunizations that you get when you're little. Tdap, tetanus, diphtheria, and pertussis. And all of those are inactivated toxins or toxoids that are administered by inoculation, and then they mount an immune response. Therefore, your immune system has been educated, so if it ever sees this toxin for real, it can respond quickly. And one of the things antibodies can do is they can bind to toxins and prevent them from binding to their target cells. So your antibodies can help protect you from these particular toxins.
I have a question.
Yes?
This might be silly, but like, is there a chance that like even if you're like teenagers and things, that like your body can still fail and like producing antibodies and you can still do it?
Well, I mean, some of the toxoids don't cause permanent immunity. And so tetanus, what do they say? Every 10 years you should get a booster. And so for some reason, sometimes the immune response to an inactivated toxin is pretty, lifelong. And in other cases, it really is kind of temporary. You really have to just, and you have to get the right immune cells stimulated in order for you to have even that 10-year immunity. So it's not completely foolproof, but very high success rate of immunization in terms of preventing these particular diseases.
What if you take two vaccines too close together? Will that increase your chances?
It shouldn't, as long as the toxin has been properly inactivated. Okay, because that toxin should not be able to harm cells in its inactivated state, but it's still antigenic, meaning it will stimulate an immune response. A final AB toxin that some of you may have heard of is cholera. This one's not produced by Clostridium. This is produced by an organism called Vibrio cholera. I don't know if you remember when we were talking about bacterial morphologies, but there were some bacteria that were a little kind of almost comma-shaped. That is the Vibrio morphology, and so that's very characteristic. Cholera is primarily transmitted through contaminated water, particularly fecal contaminated water. One of the reasons why fecal material can be such a problem in water is that some people can be asymptomatic carriers for things like cholera. And you can have this stuff actually in your GI tract. But your immune system and normal microbial antagonism is suppressing this stuff, and you don't really ever have any symptoms. But then when that is voided with the fecal material and gets into water, people who drink that water and get a good dose of this particular bacteria might not have the level of immunity that you do, and therefore might succumb to those particular symptoms when it multiplies and starts producing its toxin. This toxin is an enterotoxin, meaning it affects cells of the GI tract. And very specifically, this affects ion flow in and out of those epithelial cells. This is a pretty busy slide up here describing how this particular toxin works. The normal situation is that epithelial cells pull ions out of the lumen of the intestine and transport them into the blood, maintaining proper osmotic concentration of the blood. What this cholera toxin does is a portion of that toxin enters the cell and activates the enzyme adenylate cyclase. You remember that enzyme from physiology? Adenylate cyclase breaks down ATP into cyclic AMP. And cyclic AMP is the second messenger that can cause all kinds of physiological effects in the cell. In this particular case, the cyclic AMP affects a membrane protein, and it starts pumping those ions back out into the lumen. So we start getting all of these chloride ions and things in the lumen. And then the positive ions flow out in response to the negative gradient that's been formed. We get all these ions in the lumen. So where's water going to go now? Instead of going from the lumen into the blood, it's going to go where? Blood to the lumen. Okay? And so one of the characteristics of this is just massive diarrhea. And people can become so dehydrated that they literally die because of hypovolemic shock. Basically, your blood pressure plummets, okay, because the water is being pulled out of the circulatory system and into the lumen of the intestine. And as your water is being pulled out of the circulatory system, blood pressure is going to drop. So that is hypovolemic shock. We don't hear about cholera that much anymore in developed countries, particularly the United States, because our sanitation systems are very good. So when water is discharged, it goes through sewage treatment plants. Various processes, including chlorination, kill the bacteria that produce this toxin. But in less developed countries, where there are not good sanitation systems and people are drinking bad water, this is still a real concern. Sometimes, too, those bacteria are transferred when a ship enters port, and when they arrive, they have all this cargo that serves as ballast. What they do then, if they leave the port empty and go back to their home origin, is they fill up the hold with water from the bay to have enough ballast. Okay? And if that water is contaminated, they go back to their home country, discharge the ballast in their bay, take on more goods, and then that cholera bacteria, which is kind of unique because it can survive both fresh water and salt water, can be transmitted by ships in this particular fashion. And then finally, back to something I mentioned before. The C. diff toxin, another toxin produced by another species of Clostridium, Clostridium difficile. This particular toxin does some nasty things to the intestines. This is another enterotoxin. One part of the toxin can break down the junctions that hold together the cells that line the intestine. So things start moving across the intestine that shouldn't. And some of them kill intestinal cells directly and begin causing big lesions in the intestine. This sometimes leads to a condition called ulcerative colitis. Colitis is inflammation of the colon. And ulcerative means that colon tissue is eroding away as cells are killed. Sometimes big pseudomembranes are formed in the colon as well, like they are in the throat when we talk about diphtheria. That whole accumulation of white blood cells and dead bacteria and pus and all this nasty stuff can actually cause an intestinal blockage, which is not good. About 5% of adults normally have C. diff bacteria in their colon. But again, it's suppressed by your normal flora. About 70% of infants have it. Okay? That's something you tend to lose as your normal flora becomes more robust and more mature. When does this cause a problem? Typically, we see the greatest problems when somebody goes into the hospital and is receiving IV antibiotics. And the IV antibiotics wipe out your normal flora. Now the C. diff can proliferate and start producing its enterotoxin. And you could get all kinds of nasty conditions like ulcerative colitis. So what's the best treatment?
Replace the normal flora.
That's another possibility. Poop pills, fecal transplants, trying to get some normal flora from someone who has probably a similar flora to you, oftentimes a member of your household. Okay? But the other key in this is not just doing a poop pill or a fecal transplant. What else are you going to have to do in the hospital? What caused this problem to begin with?
Get off the antibiotics.
Yeah, you're going to have to discontinue the antibiotic, and that can be a problem because if you've had a major surgery and they're worried about infection, then that can be a huge problem. Okay? One of the things that's oftentimes associated with this is, again, severe diarrhea. One of the things you shouldn't do in treatments is to give people antidiarrheals. Okay? That really is compromising part of your immune process. Diarrhea is actually a part of your immune process. It tends to flush things out of the colon. And if you give an antidiarrheal, which inhibits the contraction of the colon, okay, peristalsis, then that stuff stays there when it should be voided instead. Audrey, did you get a chance to ask about C. diff exposure? You said that in hospital you have to be really careful and gown up and mask up and everything if a patient has C. diff?
I worked first.
Okay. Did you get a chance? Ask about that. I'm assuming it's just trying to minimize healthcare workers' exposure to these endospores so you don't contract the condition yourself. Well, that's only a few of the endotoxins. We're going to talk about a few others of them in less detail when we talk about two pretty pathogenic genera of bacteria, staph and strep. So after we finish going through a couple more virulence factors, we'll then use a couple of case studies and talk about some of the diseases of staph and strep. And we'll refer back to some additional toxins that they can produce. Okay, the other category of toxin is the endotoxin. Okay, we've been talking about exotoxins now. Remember, endotoxins are lipid A molecules associated with the LPS layer. And they're not released until the bacterial cell dies. As long as that LPS layer is intact and the lipid A molecules are part of that outer membrane of the bacterial cell wall, it's not harmful. Endotoxin poisoning causes several fairly characteristic symptoms. It affects cells of the GI tract, and nausea and vomiting oftentimes is associated with that kind of GI disruption. So is diarrhea. Fever oftentimes accompanies these particular symptoms. We'll talk about fever later on. Fever is actually another part of your body's defense mechanism against infection. Typically, these symptoms, though, are pretty self-limiting, and they're not lethal, except potentially in infants. Now, if you've ever had food poisoning before, sometimes you feel like you would wish you would die, but the symptoms typically only last about 24 hours. Fairly fast onset, usually 4 to 8 hours after ingestion of these gram-negative bacteria, like Salmonella, result in this particular condition of endotoxin poisoning. Can we immunize against food poisoning caused by endotoxin? Turns out we can't. And the reason we can't is that we will see later on that proteins are highly antigenic, meaning they stimulate a strong immune response. Lipids, like lipid A, are very, very weak antigens and really don't cause much of an immune response. So we really can't immunize against lipid A. So this is a good just summary slide here of comparing and contrasting exotoxins and endotoxins. And most of this stuff I've talked about, but I think having some of these slides interspersed help you to give some good steady tools for exam material. Okay, the last set of factors that are virulence factors are antiphagocytic factors. We haven't talked about phagocytosis yet. Phagocytosis is an important part of your body's defense systems against infection. What immune cells in your body are phagocytic? What kind of cells? White blood cells, okay, we haven't really dug into yet the different kinds of white blood cells. There are neutrophils, there are macrophages, there are dendritic cells. There are several different types of phagocytic cells. One of the things that helps to enhance phagocytosis is antibody that's bound to the bacterial cell surface. So if this is a bacterial cell, it's going to have one or more foreign molecules on its surface, antigen. It's going to have a foreign antigen on it. And if your body's immune response detects that foreign cell, then other white blood cells called lymphocytes are going to begin to produce antibody, which will bind specifically to this antigen. The relationship between an antibody and an antigen is much like the relationship between an enzyme and a substrate. It's very highly specific. Okay? Only certain antibodies will bind certain antigens. And when this antibody coats the surface of this bacterial cell, this little stem of the antibody right here binds to a receptor on a phagocytic cell. That basically allows the phagocytic cell to more easily recognize that pathogen and bind to it and engulf it and chew it up and spit it out. Phagocytosis. These antibodies that enhance phagocytosis are called opsonins. I like to say that an opsonin is kind of like the equivalent of the old junior high school, put the kick me sign on the back of someone and then watch them walk down the hall and people kick them. Okay? The person being kicked is the bacteria that has been tagged with an antibody. The kick me sign is that antibody, the opsonin. Okay, the people kicking would then be the phagocytic red blood cells.
You said the opsin is like the.
It is the antibody. It's the antibody is the opsin. Okay? An opsin is an agent that enhances phagocytosis. It makes it happen more readily. Well, we'll see later on when we talk about staph and strep that some bacteria have evolved the ability to produce surface proteins which can mask these antigens that the antibody normally bind to. There's a surface protein that some bacteria can produce called an M protein. And that kind of hides this antigen so the opsinin can't recognize it. And that means that phagocytosis then will be less effective. Another anti-phagocytic factor is a capsule. A capsule coats a cell and can cover up some of its receptors that normally an antibody or a white blood cell would recognize. And sometimes capsules also cause the cells to stick together. So sometimes clumps of bacteria form, put together by their capsules, and they become too big to become effectively phagocytized by those white blood cells. Even if a phagocytic cell engulfs a bacteria, doesn't necessarily mean it's dead. One of the steps in phagocytosis involves that vacuole that the white blood cell brings into its cytoplasm with the bacteria inside. That's called a phagosome. That phagosome then has to fuse with a lysosome before the bacteria is actually killed. That's called a phagolysosome. Well, some bacteria have evolved the ability to prevent the phagosome, the vacuole, from binding with the lysosome. So now the bacteria are inside the cell and they are protected from the immune system. So as that white blood cell moves around to other parts of the body, it carries the bacteria along for a free ride. And when inevitably that white blood cell does die, now the bacteria is released into a new location. So those are some of the kinds of virulence factors that bacteria can possess. So now let's look at some staph infections. Species of staph can contain a whole variety or a whole battery of these virulence factors. For example, they can contain multiple anti-phagocytic factors, including protein A. It's kind of like that protein M that I just talked about. Helps to prevent antibodies from binding to the surface. Staph is typically encapsulated. It also can produce that serum enzyme coagulase, causing serum proteins to precipitate on the surface of the cell, again shielding it from antibodies and phagocytosis. So it's got that. Staph can also produce a variety of exoenzymes. Hyaluronidase, kinases, the ones that allow you to bust out of the clots. Lipases. Lipases help staph to be able to metabolize skin oils, for example. They can break down those fats into glycerol and fatty acids. They can use them in their metabolism. A lot of staph bacteria cause skin infections, so that can be significant. And then staph produces a whole bunch of different types of exotoxins. Toxic shock toxin. Exfoliative toxin. What's exfoliation refer to? Removing of dead skin cells also, but in this case, this particular toxin removes live skin cells as well. It breaks down the desmosomes. Remember what desmosomes are? Yeah, they're a cell junction. They're a kind of a meshwork of protein fibers that connect one cell to another. And so exfoliative toxin can cause the skin to just kind of shed off in sheets. Not too pleasant. Several enterotoxins can be produced that affect the GI tract. I mean, I'm not particularly interested in you memorizing all of these things. What's the point here? These guys got a lot of virulence factors, don't they? Pretty impressive array. And all the different categories are represented, and there can be multiple entries in each one of those categories. That's why these things cause a number of infections. Typically, staph infections are broken down into 3 categories. And the first category are the non-invasive infections. These are primarily food intoxications. And food infections. Staph poisoning. A whole bunch of these staph infections are cutaneous infections. They infect the skin. A few good pictures to leave you with. Way up here. This is a staph infection called scalded skin syndrome. And here you can see the way that the skin literally is peeling off due to the production of that exfoliative toxin. Okay? Lots of inflammation, okay, associated with this. Impetigo. This is an infection, a staph infection that oftentimes young kids get. They have a lower level of immunity. Have you ever had impetigo?
Is that like the hand and foot?
Pardon me?
Is that like the hand and, isn't that called hand and foot? Yes. No.
Yeah, I think so. Typically it's on the upper lip or around the mouth. Very crusty. See right there? This is a boil. A lot of staph infections occur because hair follicles are infected. Okay? Hair follicles produce a lubricating substance called sebum, S-E-B-U-M. And that's kind of a waxy, smelly substance that helps the skin to be kind of pliable. And if bacteria get down into the follicle, they can then become metabolizing the sebum, lipids, and things like that, and start producing this little pocket of bacterial growth. And then the white blood cells come in and try to kill them. The staph oftentimes will produce these leukocidins, these toxins that kill white blood cells. And so we can get this pustule that oftentimes erupts of pus, and that's a boil. Okay? But it usually stays fairly localized. Acne can be caused by staph, but only about 15% of cases of acne are actually caused by staph. The other 85% are caused by a genus of bacteria called Propionibacterium. And again, a lot of times at puberty, particularly young males under the influence of testosterone, start producing a lot more lipids and a lot more sebum, and hair follicles can become infected. Okay? If we just have an infection, that's oftentimes called a whitehead, and that pus starts to form there. As cells die and necrose, then we can form a blackhead. And then if that pustule continues to become inflamed and formed, then that can cause acne. Okay? Acne is an inflammatory disease. What are the treatments? Antibiotics do help. There's an active ingredient called benzoyl peroxide, which is kind of an exfoliative agent. It cleans away some of the dead skin and helps to open up those pores, remove some of the oils. And then there is a substance called retinoic acid. That's the active ingredient in acne medicine called Accutane. And that actually inhibits sebum production. So it cuts down the production of this fatty stuff that the bacteria are metabolizing in the living room. We'll pick up there next time. Right? It's time. Talk a little bit about topical shock. So remember, tomorrow, fair amount of inoculation. I'm also going to do something at the beginning of the period. There is a computer program that runs on Windows 95. The only thing we get is run up. Called ID Bacter. And ID Bacter has a number of benefits. Number one, it's a way to study for your next lab exam. It's a way to learn some of the metabolic tests that you'll need to know to identify your unknown. It will be an assignment. You all will have to do one ID Bacter, but then you can get up to 100 optional points by doing additional ID Bacters. That's been a fairly popular activity. So I'll set up 2 or 3 computers out here you can use whenever the lab is open. You'll want to do ID Bacters. So we'll tell you how to do that tomorrow. Folks, you want to flip on the lights on your ramp?